My name is Simran and I'm a bioinformatician in the Farley Lab. Today I'm going to be showing you how to use Visualize TF sites on sequences, which is a tool that can be used to predict where transcription factor binding sites exist in a DNA sequence, and also provide information about these binding sites, including their affinity, location, and direction. Each transcription factor binding site can be assigned a value depending on the type of reference data used. TF sites is compatible with position weight matrix or PWM data, relative affinity data, and absolute affinity data. PWM data is publicly available on databases such as Jasper and Hokomoko, and has been the historically popular method of scoring binding sites, therefore making it accessible to use. We also host these datasets on the TF sites website, so you can easily integrate them into your analysis. This type of data can be used to assign each binding site with a value called a binding score. While PWMs have been extremely helpful in understanding predicted binding sites, it's known that they have a difficult time detecting low affinity sites, and their binding score does not correlate well with relative or absolute affinities. Therefore, when available, relative and absolute affinity datasets are preferable. Moving on to relative affinity data, one common example of relative affinity data that we will be using in today's tutorial is from protein binding microarrays, or PBM. PBM is a useful technique to assess binding sites because it's an empirical method of measuring the binding between a transcription factor and DNA sequence. This data is accessible on the UniProbe database, and we also host it on TF sites. This type of data can be used to assign each binding site with a value called a relative affinity. And importantly, PBM correlates with methods that measure absolute binding affinities, which is our last category, and this includes techniques like Mitomi or BETseq. These methods can also be used as reference datasets in this tool to measure the affinities of transcription factor binding sites. In this tutorial, we will be showing two examples. The first is a known search of transcription factors. In this example, we'll be looking at the ZRS enhancer, which drives expression of the Sonic Hedgehog gene in the developing limb. We will search for ETS1, HOXA13, LHX2, HAN2, and PBX transcription factor binding sites. All these transcription factors are known to regulate the ZRS. From functional studies and crystal structure of these transcription factors, we have determined the minimal core site for binding and can use a PBM dataset when available to annotate affinities. The second example that we'll be showing is an agnostic search for transcription factors, which is more of a general search where we don't necessarily know which transcription factors to look for. We're again annotating the ZRS enhancer, but we'll be using a list of limb transcription factors. And we'll be using different types of reference datasets to show how this tool can be compatible with various data types. To run this tool, the first input that you'll need is a list of one or more DNA sequences to annotate. So we'll be using the ZRS enhancer. You can manually enter its sequence and its name here. And then we can grab its sequence and paste it here. And then if you have more sequences to add, you can continue to add more rows, but we just have the one. Or alternatively, you can also upload a file that contains the same information. So that's what we've shown here is a file where one column is the name of the sequence and the second is the actual sequence. So we can drag that here. Next, you'll need to provide information about the transcription factors that you'd like to analyze. For each transcription factor, the minimal information that needs to be included is the name, the color that it'll be assigned on the output visualization, and its binding site definition. The list of possible colors can be found at this link here. Optionally, you can include the affinity reference data file used to assign each binding site a value. And if you want to normalize an affinity dataset, you can use the Normalized TF-DNA Affinity tool, which is linked here at Created Affinity Dataset. The output files from that tool can then be used as input files here, and they'll be automatically available under the My Affinity Dataset dropdown. You can also enter a minimum affinity needed to predict a site. So this information can be manually shown here. So for example, the first transcription factor we want to look at is ETS. So say we want to label it blue, and then we have its core binding site definition. We have already created an affinity dataset for ETS using the normalized affinity tool, so that'll be available here under this dropdown. And then we're not going to include a minimum affinity for this example, but you can continue to add more transcription factors by clicking Add Row, or you can upload a file containing this information. And that file would look like this, where again we have the name, color, binding site definition, and the reference data, and optionally the minimum affinity. Each row represents a transcription factor with its corresponding information. For ETS, HOX, LHX, and PBX, we will provide PBM data, but for hand we only include the binding site because there's no PBM data available for it. If you're using any affinity reference data, the name of the files referred to in the TF information file, shown here in this column, must be uploaded here under TF affinity reference files. So first we want to upload that table we just showed, and then all of our PBM data that we are using. If you would like to analyze transcription factors using PWM data, then you can do so under this batch motif information parameter. You can either use the default motif databases that are hosted on the website, or you can use a database that was generated using the Generate Motif database tool, and those will show up under here, under My Motif Databases, or you can upload a custom file. So for this example, we'll be using PWM data for the PBX2 transcription factor to score its predicted binding sites, and we'll be uploading a file for that, which we have shown here. And there are a few other parameters that you can choose to specify. The first is the PWM minimum score, which is the score required to predict a binding site. By default, this value will be 0.7. And then all binding sites scored by PWMs will be colored gray by default, which can also be changed. The other remaining inputs are optional specifications for the output visualization. The output images will automatically be in PNG format, but you can choose to additionally output the same images in SVG. The resolution of the plot can be adjusted as well. By default, it will be 200 pixels per inch. And if you'd like to look at a specific portion of a sequence, you can do so using the zoom range parameter. You can specify the start and end coordinates for the region that you'd like to zoom into. And so we're going to leave these as their default values. And now that we have all our inputs in, we can go ahead and hit submit. While the job is running, you'll see this page, and the job will be assigned a number, and then we can come back when it's finished to see what the output will look like. So now that the job has finished running, we can see the outputs that have been produced. There will be two types of outputs. The first output type will be images that visualize the predicted binding sites across the DNA sequence. So we can zoom in to our image and we can look around different regions at our binding sites. And this first image here is the full sequence, and then these next two images that follow are each of the sequences broken up into 500 base pair windows. And you can also download all of the output files here as a zip folder, and this is what it looks like when you download them. We have the images that we just looked at along with each of the images in PNG and SVG format, since we specified that in our inputs. And then we have the second output type, which include TSV files that contain a list of all predicted binding sites for each sequence to be analyzed. And there will be two separate files if you're using PWM and affinity data. So the first will contain information about each binding site for the sites that were annotated using PWMs. So that is shown here. And then the same information will be shown for the sites that were annotated using affinity. In our second example, we'd like to analyze other transcription factors that might regulate the ZRS enhancer. We don't know which exact transcription factors to use, so we'll be annotating a subset of transcription factors that are co-expressed with a gene regulated by the ZRS enhancer. For the first input, we will use the same as our first example, with the sequence to annotate being the ZRS enhancer. We're just going to upload that as a file. Since we don't have any affinity data for this example, we're going to leave this section blank, and we're going to be using PWM data to score transcription factor binding sites. We've already used the Generate Motif database tool to filter for the TFs that we wanted, and the resulting file can be accessed under the My Motif Databases dropdown. We're going to set the minimum PWM score to 0.85 to set a stricter threshold, and then we're going to leave the default color of the binding sites as gray. we're also going to choose to output the images as SVGs and leave the default values for the rest of the parameters, including plot resolution and zoom range. So now that we have all of our inputs in, we're going to go ahead and hit submit. And while the job is running, we'll see this page, and then we'll come back when the job is finished and we can see what the output looks like. For our output, we get images of the sequence, again, the full sequence, and then divide it up into 500 base pair windows, and we can zoom in and look at our binding sites at different regions. And then when we download our zip folder, we also get the table containing all the binding sites and information about them. So that concludes this tutorial, and thanks for using TF Sites, and good luck on your research.